Hello, my name is Fran Romero, and this afternoon I'm going to talk about our poster number 32, entitled Multiomic Characterization of Diurnal and Seasonal Cycles in the Marine Microalgae Ostreococcus tauri. We have chosen this microalgae because of its simplicity. It has fewer than 8,000 proteins, and it is located at the evolutionary base of the Greek lineage. To study diurnal and seasonal cycles, we cultivated Ostreococcus under long day and short day conditions, and we also included data under free running conditions consisting of continuous light and continuous darkness. Based on our RNA-seq data, we found that almost 80% of the transcriptome present rhythmicity under LD and SD conditions, but when our free running conditions were included, we found a more complex picture where only 25% of the transcriptome is bona fide circadian, presenting rhythmic expression patterns under continuous light and continuous darkness, and both photoperiods. Based also on clustering analysis, we found a set of genes that were either light repressed and dark activated, or the other way around, and these only presented rhythmicity under dark and light alternating regimes. We found that the effect of photoperiod shortening produces changes in the phase and also a reduction in amplitude. But more interestingly, we found the emergence of complex rhythmic patterns with two peaks during the day, like this one here. But when these genes were studied under continuous light or continuous darkness, we found that only one peak was observed. Applying fast Fourier transform, we found that actually this complex pattern emerges as the combination of two simple rhythms, one induced by the light photoperiod and the other one induced by the dark period or scotoperiod. This allowed us to propose different transcriptional programs under LD and SD conditions. For instance, under SD conditions, we found that nitrogen assimilation genes peak early during the night, which does not make much sense. So we decided to study the proteome. We generated proteomic data covering more than 40% of the proteome, and we found a drastic reduction in rhythmicity. Like here, we have a very nice rhythmic transcript and a flat, noisy protein. Only 25% of the detected proteome was rhythmic, and for these rhythmic proteins, we found an offset or a delay between the peak of the transcript and the peak of the protein. That was very clear in the S-phase genes in the cell cycle, where you have an offset between transcript, proteins, and the actual number of cells in the S-phase. These translational offsets depend on each protein, but do not change between LD and SD conditions. So in order to make sure that the proteins are present at the right time, Ostreococcus changes its transcriptional program. And this explains why nitrogen assimilation genes peak early during the night in short day conditions, just to make sure that the proteins are available at the very beginning of the light period. Since the transcriptional program seems to be very important, we have generated ChIP-seq data to study the main regulators of gene transcription, epigenetic marks and transcription factors. And we have found that although the epigenetic marks do not change over time, we were able to identify that starch biosynthesis and cell cycle genes are target of OSTA-CCA1 besides OSTA-TOP1.